Well, welcome back to part two of how a plant grows, a plant's perspective. I just want to make a couple of comments about the last one that I thought might be of interest. Uh, when we talked about that electron transport and photosynthesis, I forgot to mention there are a number of herbicides based on their mode of action actually target either photosystem 1 or photosystem 2 or the electron transport. Just so you know that when you see some symptoms, if you go to the modes of action of them, you'll know which ones are affecting those transport systems. And I guess the one thing I took out, if there's anybody that's interested, is besides serotonin, dopamine can be affected, and all basically chemistry in the human body can be affected by nutrients. And Dr. Kaplan has just published a new book, I believe, and if that interests you, just send a note to Taurus, and we'll see if we can get that for you if you're interested. But today's presentation, if you survived the last one, and that for me was a very difficult one because it's really complicated, and I still haven't figured out a way to. I can make it simpler, but leaving out a whole bunch of steps, but basically, like I said, you don't see them. You just need to know that those reactions are taking place, and it's the nutrient complement in the cell and in the chloroplast that's really key. But we talked about water and how important it is, and I'm going to make an effort to talk about how water is taken up. This isn't my specialty. Roots and shoots are, and sugar, and how plants take up nutrients and water and move it around. Now we know this, that water drives everything. That transpiration flow of water from the roots and out the leaves is what really keeps the system going. And without water, the plants shut down. And that leads to a lot of stress, which I'll cover in the next presentation. But just to summarize, here's what happens is you get water molecules coming in, goes up the xylem, as we talked about, just a tiny review, up into here and out the stomata. And that's both cools the plant and also gets rid of the oxygen through those stomata. So it's really, really important process. And as I said this before, this is what happens when that flow shuts down. And when this occurs, the plant has to shut those stomata to conserve water. So what's water? Water is kind of interesting because the molecules all stick together. That's why you can pour it and it'll stick together. And that's why when it starts pulling in a vacuum, like you see here, is sort of like a siphon, the water pressure or water potential, because there's lots of it here, is great. The water potential here is low because there's not as much because it's losing it. So that in itself, along with adhesion of the water molecules because of these hydrogen atoms attaching, hydrogen bonding with the oxygens, keeps them stuck together. And that's why if you sort of spray on a waxy surface, it'll form a bubble, a little blob of water that sticks together. And that's why we use surfactants to get it to run apart, because it wants to stick together and flow. And that is part of basically how that transpiration system works. But it's really kind of cool. Now, what is water use efficiency? And everybody says, well, how much water it takes to grow a bushel of whatever. And here are some of that. Like it takes 41,000 pounds of water to produce a bushel of canola, 30,000 pounds for a bushel of wheat, and 37,000 pounds for peas, and 16,000 for barley. Now, there might be a clue for you there. Barley doesn't use as much. Now, I don't know if it's as tolerant to drought, but it doesn't use nearly as much water to grow a bushel. And you would think that's crazy, except for the fact that almost all of it is used in transpiration to cool the plant to move nutrients, and the plant doesn't keep much of it. So that's why the numbers are bizarre. But what is really efficiency? Well, I consider it, and we talk about this because it always goes back to photosynthesis and carbon. If photosynthesis shuts down or is slow, the plant can't produce as much anything, because that's the secret. So if we have plants that use, it takes 400 molecules of water to process 1 molecule of CO2, that's an efficiency that's very low. Well, what if we had plants that only took 150 molecules of water to process 1 molecule of CO2? That would be really good. And unfortunately, all the plants we grow up here are this top part here, which are called C3 plants. The majority of them are, except for one. What's this 150 molecules of water? Which plant is that? Well, that's a C4 plant. That's corn. That's corn for us. Corn is very efficient because of the way it does its photosynthesis. That's what the secret is. So people are now trying to make C3 plants into C4 plants, and if successful, that would be a huge, huge Nobel Prize, I'm sure. But anyhow, I'm thinking this took a long time to evolve, and I don't think that's going to happen. But anyhow, it is kind of interesting, and we'll that's why we're going to have corn growing all over the prairies potentially with time, because as it gets warmer, if it does, and drier, it will do better. Now, you need to understand one thing, and that's the term osmosis, is the movement of a solvent across a semi-permeable membrane towards a higher concentration of solute. And the solute can be salts, can be sugars, and so on. Those are solutes. So if you've got a high concentration of a liquid, I'll just show you a picture in a minute. Now, and that'll help explain. And osmosis is responsible for the ability of plant roots to draw water from the soil, because there's a membrane. Remember that plasma membrane? There's a membrane there, so it's got to move through that membrane, and I'll show you how that works. Plants concentrate solutes in their root cells by active transport. They pump those solutes in because in many cases, the actual nutrients inside the root is higher, so they actually have to actively pump it in through the active transporters. And water enters the roots by osmosis. Osmosis is also responsible for controlling the movement of guard cells, and that's partially true. But let's look at that. So if you have water, pure water, and you have water with a sugar or salt in it, the water will flow from here into here. It will move from a high concentration of water to a low concentration of water. So when you have more solvent or solute in there, sorry, you have less water, and it will move that way. And I'll talk about that, but that's really key. So let's leave that for a minute, and it'll pop in later on when we look at how the roots work. So water in the plant and the system follows the growth curve. It is this uptake of water up until flowering, where the plant is building all this biomass and building all the reproductive parts, and then it dwindles as the plant matures, and we're using a reduction in the amount of zinc here in the roots, and we're making now the zinc up here in the seed. So when the zincs change, because up until flowering, the zinc are of course the roots and the shoots and the leaves and all of that kind of stuff. But once flowering starts, now the zinc is the seed. So the plant has to make a decision. It reduces the amount of nutrient going to the roots, and therefore the root effectiveness becomes less. The efficiency becomes less in extraction, and depending on the deficiency or the lack of nutrients or the timing, it will start pulling the nutrients from the roots and from the bottom leaves up until here, and then it dwindles off as it ripens, because once the seed is full, we got maturity, and then it starts giving off water to dry down. So here's where it starts, and I'll show you this. I'm not sure the sequence is totally right, but I need to start here. So here's a wheat root, and you can see this is the xylem, the big holes, and then the phloem, because the xylem is dead tissue in here, and then around there's the tissue of the plant. And this is the rhizosphere. This is where the plant root hairs and the mycorrhizae may be attaching things and sticking all the stuff to the root. Then as we move up, we get the phloem is full of sugars, and here's the xylem, which is moving water up, and the phloem is moving in photosynthates or sugars up and down. As it moves up, it'll move into lateral roots, and you can see this is soybeans. It's a nice stem to work with. You can see that there's a pith here in the center, and then you got xylem and phloem mixed all through this cambium layer here. Here's wheat, and the same thing. You can see wheat, of course, hollow stemmed in this particular case, and then you got the xylem and phloem that is moving in here to do that. So it keeps moving up, and then when you hit the leaves, I cut this. This is soybean. I kind of like working with soybeans. They got nice roots and nice leaves. So when you cut through the leaf here right at the base, and this is the primary vein going in, and then here are some of the minor veins, you can see that it's got the xylem and phloem in here. And focusing, and you can see the xylem's up here and the phloem's on the bottom in soybeans, and then all the smaller veins just have a smaller form of it. And then here's wheat. You can see that again, the xylem are the big ones, and the phloem are the small ones around there. And then they're in the longitudinal veins in the leaf. We have the same thing. So what I'm getting at, there is a continuous tube that runs all the way from basically almost just behind the root tip, all the way up the leaves, up the stem, into the leaves, into the venules, right through to the tip of the leaf. And if there's too much water left there, sometimes it will just ooze it out at the tip and dump it. Okay, you can see that from time to time. And the water is leaving, and here's what happens. As the water leaves, the water goes into these spongy cells with a lot of space in it, and the water becomes vapor. And that allows it to then leave once the stomata open, because if we have a low water vapor content outside the leaf, the water will, or water vapor will move out, because the water potential out here is lower than it is in here. There's a lowering water potential all the way from the root to the shoot and to the stomata. And then CO2 can go in here, and the water can go out there along with oxygen. So that's all there is to it. It's just evaporation or transpiration with water pressure from the bottom and pull from the top. When this shuts down, that's when the problems start. So let's see how this works. And I have to do this in two parts, and I continually have to apologize for trying to give you these diagrams, which may look complicated, but I just need this to show you some of the names of things in here so you understand how it works. Now, the one thing about the stomata on the upper and lower parts of the leaves, they have their own chloroplasts. So they can make ATP independent of the rest of the plant. The stomata are so important to regulating water loss that the plant has isolated them and allowed them to make their own ATP. So when we have stimulation, we have these transporters, as I talked before, and you can see this one is a transporter for chloride. And there are some cells here called subsidiary cells that are supplying all these chemicals and nutrients basically into the plant. So we get chloride being pumped in, we get potassium being pumped in, and gates. And there's channels. Here it is. There it is. Potassium channels, and we got proton pumps that are pumping out hydrogen, but we're pumping in very, very quickly high levels of potassium, and we're putting in some nitrogen. And look what happens. The water flows in. What did we say? When we increase the level of solutes inside of a membrane, which is potassium and chloride, when you notice that the chloride is negative, positive, it balances out, and then we got starch and sugars and malate in here. The water flows in. The water flows in, brings about osmotic pressure, and opens the stomata. Okay. Now let's take a look at this, and this is just a quick diagram. This is early morning, and blue light strikes the guard cells around the stomata. That's closed. Potassium and chloride and hydrogen move in. That then will cause the water to move in, pressurize the guard cell, and it pulls it open. So if it wants to close, what it does, it kicks out the chloride, it pumps out through gates very, very quickly the potassium, reduces the solute level, the water follows, and the stomata close. So it's a function of, it's driven by potassium and chloride, and abscisic acid is involved in controlling this for sure. So it's just a movement of solutes, which are potassium and chloride and some calcium as well in there, in and out, that's followed by water or leaving of water. And that's the beauty of potassium. Potassium is so important in osmotic pressure and turgor pressure in all the cells, and is used for that purpose. So let's take a look at how that reflects, what that means for the plant. And this is timing, 7 in the morning, 11, 5, and midnight. Remember I said it showed it, sunlight, blue light basically, early morning blue light hits the guard cells. That stimulates the opening or the chlorophyll to start producing energy. So here's where we go. Let's take a look at stomatal opening. Well, let's look at potassium. So early in the morning, sun comes up. It starts opening up. Potassium starts moving, starts opening up the stomata. The potassium basically rises to its max. The stomata open to the maximum by about 1 o'clock. The potassium now starts decreasing because the stomata are staying open because it's now photosynthesizing full tilt and producing sugar. So by 11 a.m., it's full tilt. Pressure is on. It is going full tilt. By 5 o'clock, it's starting to decrease. Your sugar levels are decreasing along with your aperture, and then by midnight, you're back to a sort of normal pressure. And that's how it works. And it's stimulated by the sun coming up and activating the chloroplast, producing energy to allow the plant to pump in the potassium, which is followed by water, which maintains that opening, and the water keeps it open, and it runs through the day. So what happens when it gets cloudy? What happens when it gets smoky? What happens when it rains for a week and every day it's cloudy? You know what happens? The plant doesn't go anywhere because it can't produce enough sugar to, in fact, just keep the cells alive, because it's got to use some of that sugar every day to keep the cells alive, night and day. So it doesn't use all the sugar and it doesn't store all the sugar. It keeps it in a transitory form to use at night. So your plants can actually stand still for days under those kinds of conditions. That's just how the plant manages that. And here is stomata. This is wheat. You can see there's just all these little white specks you see on the bottom of the leaf are stomata. So there's like thousands to millions of them. But you can see here when you get diseases on the leaves. What do you think that does to the stomata, the flow of nutrients along that vein? Another one of those things that leads to that reduced yield and increasing that yield gap. And we have pretty good controls now with fungicides. And here's just another shot of them, a close-up. These are really small because my microscope can't get any bigger than that. And you can see that there's that opening and it's closed because I cut that leaf off. But these are all stomata on the leaf. Now, looking at the leaves, but let's look at the roots. What happens to the roots when we get dry? Well, what happens, and to me this looks like a soybean root because there's a nodule there. What happens is during drought, the length increases and we reduce the lateral roots. The length increases. The other thing it does, and I think I mentioned in my last one, is when it hits compaction, it just thickens the roots to try and penetrate through rather than having a spindly one. So let's move on to the roots so we can understand that aspect of the roots and how they take in that nutrient. And we talked about the plant genetically being able to influence where it puts lateral roots and so on, but we'll just show you how that happens. I'll just go back here. So, I mentioned the shovel right up front. That's why you have a shovel, because most of your plant is underground. If you're not looking at what that's doing, you're getting only a top glimpse of what where the real action is. So here's how plants in a general scheme take up nutrients. Here we got the root hairs, and there's two things. They can pump out that hydrogen, and we've seen that we got hydrogen pumps. that we get out of water. And we also put out in the roots, because the root can't feed itself, but it has to respire. So it takes in oxygen in the roots and it produces CO2 in respiration, like all living creatures. That is kicked out and it reacts with water to produce carbonic acid, which gives you hydrogens, which can knock off the cations on the cation exchange, because the clay particle, organic matter particle, is negative. These are positive. So if you flood it with more positives, it'll kick them off, and the plant can then take them in through the other root hairs. So you can see off the tip here where we got a lot of respiration where the plant is going, so we can knock those off, and that's basically to some degree how the plant can manage its cation exchange. You know, and I'm not a soil kind of guy, to tell you honest truth, and you know, I appreciate a little bit soil scientists, but I'm not one of them. So let's take a look at roots, because people always say, well, how long are roots, or how deep do they go? And I always say they go as far as they have to, because if you think about the source-sink relationship, if a plant is forced to go deep to get water, that means the shoot has to feed it to do that. And it's losing the ability to extract nutrients, but it's got to go for water, because without water, you don't get transpiration in the flow of anything, and the plant can't cool itself under hot conditions. You know? So when drought happens, the shoot suffers even more, because it's got to keep the root going and keep it alive, or else it's doomed. So let's look at roots. And somebody actually figured this out. Kilometers of root per meter squared. So there's 5.3 kilometers in barley. I didn't point chickpeas, but you can take a look at one thing. Legumes have puny roots. Here's chickpeas at 4.5. Faba beans, that monstrous plant, with only 2.9. Maize has got a pretty good root. Spuds, so-so, doesn't go down real deep. But look at canola. This is old grape. This was done in '89, so that would have been, I think, in Europe, lowland, the Philippines, that was done. Oh, in UK, sorry, in Europe, so I thought so. So 28. Interesting, isn't it? I'll get back to that. Oops, I'll get back to that in a minute. Soybeans, 5.5, and wheat, 1823. Maybe we can understand why we grow a lot of canola and a lot of wheat, because they got great roots. Canola especially, because canola, or the crucifers as a whole, are different than the rest of the crops we grow, except for quinoa and beets, which are chenopodiums. The crucifers and chenopodium never had mycorrhizal relationships, and maybe never will. We'll see. So they had to develop their own system of extracting nutrients and became very good at it. Very good at it. Wheat also is great. On our prairie situation, where we have very traumatic kinds of events, that's why wheat and canola grow here in large amounts, and that's why things like peas and lentils and well, lentils pretty good, but peas and other legumes don't do as well. Their root systems are not nearly as aggressive. And faba beans and soybeans don't take drought worth a hoot. But let's take a look at what was done here in the prairies. And this just came out in Top Crop as well, and I'm not going to go over everything, but let's take a look at our good friend. Where's Mr. Canola here? Right here, canola. D50 means that the density of roots for 50% of the roots in the plant all are within 8.4 centimeters of the top. Man, those big roots are on the top where there's lots of nutrients. They can extract nutrients. A little bit of rain and those roots are right there pulling it in. Soybeans, you know, and it goes down fairly deep, but not as nearly deep as soybeans. And soybeans have 50% of the roots in the top, 10. But on the whole, most of the roots, 50% of the roots in the top 6 inches. 95%, go down a lot more, and then for 100%, they go down a lot more. But in canola, that's a lot of expenditure to go down, to get those extra few roots, another 5% of roots, and they only do that if they have to. And that's all the point is there. Look at peas. They got a little deeper, but you know, they're fairly shallow on the majority of their roots. They will go down, but you know, not greatly. Barley is the same way. Most of these are on the top, you know, 10, 12 centimeters, less than 6 inches is the majority of the roots. So if you're getting compaction at 4 inches or 5 inches, you know, that's affecting you a bit. If you're getting compaction at 2 and 3 inches, then, you know, at the driveway to your fields, you get that. Well, that's why you got no crop growth, because those roots can't go down. Now let's take a look at a root. And I do a diagram because, and I'll show you this in reality, but I can't show you everything. Key thing is here, this little gray spot is where the plant makes new cells. The yellow part is called the pericycle, and that's key to understanding what's going to take place in terms of growth of lateral roots and rhizobium, how they infect them. Now you can see that the plant pumps down stuff of photosynthase, not stuff, photosynthase down into this zone to feed the new developing embryos here of the cells, and also the covering the root tip here. And then whatever's excess flows up here near the epidermis and back up to the top. So let's take a look at a real root. Here's a wheat root, showing you the tip is bare. It's kind of open because it's got sensors of all kinds in there in this plasma membrane to sense what's going on: pressure, nutrient levels, water levels, oxygen levels. So it's open to its environment. It's covered by a little cap. And then there's an elongation zone, because if the plant is producing these new cells here, it uses hydraulic pressure to expand this root through here and extend the length of the roots. Oops, go back. After the elongation zone here, the root hairs start developing. They don't start here because they'd get ripped off if it's going through the soil. But also right here, the plant is putting out all these exudates, sugars primarily, amino acids, which feed the microbes. So as this root goes through the ground, and these root hairs start growing here where this plant put off its mucilage here, microbes develop, and the microbes are after the carbon. So that's the activity of the microbes breaks down minerals in the organic matter and in the soil, defecates them out, and they become available to the plant and extracted by the root hairs, which pick up about 60 to 70% of all the nutrients are picked up through the root hairs. Further back, the plant then puts out lateral roots to expand this rooting zone. But these root hairs live 5, 6 days, 7 days, and the plant has to be growing through the soil continuously to access new minerals and water. So here's just a closer look. You can see that this is all covered with cells that are going to just slough off and are all sugary. Here's the rhizosphere I showed you before. That's where the plant gets most of its nutrients. And I'm a believer that this plant is putting out carbons and sugars and proteins that influences which organisms it attracts. Because if you take this soil around the rhizosphere, that zone right around the root, the spectrum of microbes in there will be different than the bulk soil. So it is influencing what it wants there. And this is just mucilage around the tip, you know, which is largely sugars and so on that feed those microbes, and also help break down the inorganic or the organic into inorganic as well, after things like citric acid, malic acid, mugenic acids, and those kinds of extractants that the plant uses. You know, and the fact is we're just starting to learn about all these organisms and in that top 15 centimeters, but we'll learn a lot more, and meetings are now all about microbes, so you're going to be bombarded with all kinds of stuff. Now roots grow at various angles. You can see here in wheat, it grows at a 40, 38, 40, 45 degree angle very consistently. Amazing. They don't grow just randomly, they're all consistent. And so here's basically how water enters. It comes in through the root hair, and it goes through these plasmodesmata, or it goes along the plasma membrane. I'll show you a better shot in a minute. And it goes from here, and this is impassable. So the nutrients that are in between the cells has to go into the cell. Remember what I said? You gotta, if you're gonna deal with homeostasis in the cell, you gotta be able to control what's coming in and what's going out. So it's forced to go in through transporters through the endodermis into that pericycle, that little part right next to the xylem, and then through that into the xylem. This is perhaps a better diagram of that. So the root hair, which is one cell, notice that. So if you kill off a root hair, it's not a real big deal. It's only one cell. It dies very quickly and puts out more very quickly, and it feeds. There's a big vacuole here, and it feeds as it's absorbing, and it's getting, some nutrients fed back to it. So you can get water going in between the cells, but eventually it's got to go in to the cell here to get past this Casparian strip, and then once it's inside of there, it's into the xylem, and away she goes. So this is what roots look like. Wheat has 5 seminal roots here that come off the seed, and then after that we get crown roots that develop just below the soil surface, which broaden out and allow it to feed over a bigger area. And we'll just show you some roots here. I'm sure you've seen them. I always like these because you don't have to know where the mid-row band is with canola, because just look at the roots in the fall time and it'll tell you, because the roots are picking up nutrients in the bloom of the nitrate and ammonium that may be there. It can pick it up as those mobile nutrients from mid-row or sideband move out into the soil. The roots will detect it and then can influence the growth pattern and the production of lateral roots. Look, they put all these lateral roots because they know there's a food source there. Same thing here. There's 3 lateral roots going over to the mid-row band. So that's the beauty of these plants. They can detect these sources, and that's why fertilization in a band or in a mid-row band will eventually be found, because there's a big bloom of nutrient that the root tips can detect. Here's wheat. You can see there's a lot more fibrous stuff, and it's very good at scavenging near the surface, which is where we have the nutrients. Here's soybeans. It's got a pretty nice rooting zone right up in the upper layers, but it can go deep. And you can notice that almost all of these, once you get down into past that 8-10 inches, the lateral roots are very minimal. And at that point in time, the plant is not going to spend more of its photosynthase, its sugar from photosynthesis, on this root unless it has to go down for water. It'll just put them up here, grow more root hairs. And we know that, different plants go down deep, and we know that alfalfa goes deep, and you want to have deep-rooted plants because those old root lines, when they rot out, are used for water to move down and also for other plants to follow those roots down. So if you're going to do some ripping, for example, you might want to follow it up with a crop that's got a really deep rooting system, and as big a rooting system, or a number of plants with rooting system, to establish those root channels down through any compaction that may take place, and also put down some nutrients down under that compaction layer or right out, so plants can have access to nutrients at a deeper level. And we got to provide those nutrients, all of them. See this, the biomass is growing. Well, you need NPKs, and we need all the microbes, but we focus generally on these because these build the cells. The N and the P build the cells. The potassium drives it. The S is involved in a whole bunch of things, including the building of the proteins, because nitrogen and sulfur are the key to building proteins in the plant. Let's take a look at lateral roots now. Here's that diagram again, and I want to show you how the plant determines where to put lateral roots based on the signals and the hormones, and I'll just show you that. But here's the plant, and here's this pericycle right here, this little yellow line, a single cell, and it's right up against the steel. So here's a faba bean plant I lucked out one day when I was skinning it. You can see that the lateral roots are forming, and this one hasn't even shown up at the surface. This is just starting to pop out a bit, but it starts right here at this pericycle. It's kind of like a stem cell, so it can form it. And here's how it works. This is for really the guys who are caners, the young guys, some of the older guys may have some difficulty with following some of this, but since you never see it, it really doesn't matter. It's just happening. You just know it's happening. So auxins, cytokinins are matching off and putting a stop and saying, oh, I think we need a lateral root here, and abscisic acid also is involved all the time. So it starts forming and dividing in what they're calling this the pericycle, which is true, and then they call them founder cells, I call them stem cells. And and then the auxin and cytokinins we get developing and bulging out, and then the auxin, ABA, and cytokinin control this formation, and poof, you got lateral root. So you got lateral roots, and you got all these root hairs, and there's your lateral roots, and the plant is deciding. You see, some are going that way, some are going that way on various sides of the root. And the lateral root, if you look at it, you can see that the xylem and phloem actually run all the way back in. So once it gets in there, it automatically goes to the xylem. So there's no holding it up once it enters here. And you can see this has got its own apical meristem now as a lateral root. And there it's coming, and you can see it's opening up very nicely. The plant just loosens the cells to allow that root hair to move out. Keeps moving, poof. There it is, it's popped out. Now let's take a look back. So if you cut through it, you can see that now that that lateral root is out, it actually goes right back here, and you can see all the xylem and phloem moving and hooking into the primary root channel. And away she goes up and down. Now let's take a look at root hairs more closely. I just want to show you some stuff here. You can see that these are peas, and you can see this is one of Taurus's product, polysulfate. You can see that the actual root hairs will dissolve the rock. This is poly in wheat. Same thing, you can see there was a little bit of toxicity. I'm not sure from what. If you see that browning, there's something going on, but you can see that there's no damage here. It's actually cutting a groove right into the rock, pouring root hairs into it. Same kind of thing. You're showing the root hairs will grow and try and get access to that. It's even putting out a lateral root here to try and hit it. This is a tip of canola growing right along a granule. And I like to see this because the tip is right up against it. And if there's toxicity, you're going to see it at the tip. But you can see that there's not much here. And then you can see the rooting, the xylem system and the internal system of the plant to take those nutrients back, and then the root hairs are spreading out and digging right into those granules. I just want to talk about fertilizer effect for a minute, because this is still a concern for people. And this is the index, salt index. So you take a look at ammonium sulfate 2124. It's got a salt index of 88. The higher it is, the more salty it is. There's sulfur plus at 5, and there's potassium chloride at 120. What did I say about salt in water? If you have lots of salt in water, the higher water concentration will move to it. So let's take a look at what happens. I'm potassium chloride right there. Okay. Now here is ammonium sulfate on the left. I just did this, a couple of months ago, just for fun. And you can see it. It's almost clear. It's like glass. It's just a crystal. And this is potassium chloride, or muriate of potash, whichever way you want to look at it. And it's a crystal too. It's a salt. So here's what I did. I took them, and here, as you can see, the last bit of a granule of ammonium sulfate. And so I took a bunch of products and I put them down. The big one, a medium one, and a small one. And this is potassium chloride. And this was ammonium sulfate after 42 hours. Basically, the ammonium sulfate is gone. This is what the potassium chloride looked like. The big granule was left, but you can see that basically the whole granule has disappeared, and all there's left is a little bit of this orange color that gives it the color. So what happens is that ammonium sulfate and potassium chloride with the salt index absorb water very quickly, So therefore, it allows the water to react, and it releases the potassium, in the case of potassium chloride, and the chloride very, very quickly. And the same with the ammonium sulfate. And ammonium, it can be very toxic, but it also absorbs all the water from around it because it's such a high salt content that the water is just sucked right up. Same with urea. That's why urea granules disappear very quickly. It's a high salt index, and it just sucks up the water. So if you put too much into the seed row, and there are limits on this, it's a great product. All of them are great products. It's just that you want to be careful not to use too darn much of it, and especially if it's dry, especially if it's in the seed row, because the closer it is to the seed, the more water it pulls out, and the less water is there for imbibing by the seed to germinate. And really, the salt index to me is a problem because of the speed at which the reaction occurs and the flush of ammonium and the drop in pH and the sort of toxicity related to that, as well as the potassium chloride. The speed at which it happens is the issue. So if you have it in the side band or even in the mid-row band, and the potassium doesn't move anywhere because it's not that mobile in the soil, or an ammonium, well, it'll be converted to nitrate, and then it can move, but the plant roots will find it. Just keep any large amounts away from the seed in close proximity to the seed to reduce toxicity and to reduce the delay in emergence for that. It's just the way it is. They're great products in the right place, but don't overdose them or be cognizant of the risk. Just showing you some more. This is crystal green. And here's gypsum. You can see the same thing. Plants like gypsum, and you can see it cutting holes in there. Here's 1620. And you can see this isn't too bad. That's not too bad a dosage. I'm surprised. But here's a cool thing. You see it burnt off all the root hairs, but it didn't damage the stem. And once the root got by it, it just popped up more root hairs. So the plant can control that. Here's one that you want to be cautious of. And this is 10% boron granules with canola. Boron is, you got to be careful how you use boron. Boron is a foliar, or if you put it as a spray in the furrow and dilute it down, is okay. That's not bad. We have it in various seed dressings. The big risk, as far as I'm concerned, from what I've seen, is if you got a large granule with a high concentration in the seed row. I'm very leery about putting any granular boron of high concentration in the seed row. It's a choice you can make, but there is a risk. And I don't even see why, because you can broadcast boron. It's an anion. I'll let water take it in, and I put down as high as, and I don't recommend this, but for trial purposes, I put down as high as 10 pounds active boron right the day after seeding on canola with no signs of toxicity, and that all depends on moisture. But if you're going to use boron, and because there seems to be such an issue with deficiency, I would, on your own land, start out by if you're going to broadcast or you're going to do anything, start off by putting in some strips of variable rate. Start at a pound. I think one of the issues with why a lot of the research on boron doesn't show much for results is that they're using foliars or very low dosages, and you can't do much with that. If you're going to try and fix boron, you're going to have to be looking at a couple of pounds to 5 pounds an acre, in my opinion. And I'm not an agronomist, so don't hold me to that. But be cautious with the granules and do your own trials on your own farm. Here's S15. Canola loves this stuff. Anything with sulfur, canola loves. This is sulfur plus or gypsum. Same thing. The root hairs, when you see roots that aren't damaged, you know that you're in good shape. Here's my photograph of canola burrowing into struvite, or what was crystal green, and you can see that the plant once it found some, it started putting out more lateral roots and more root hairs and actually burrowed right in and dissolved with citric and malic acid, dissolve that phosphorus to get it. Plants are smart. This one was going towards urea, got a heavy dose, turned around, ran away. It got away in time. A couple things on that shovel I talked about. You can see here that in spite of how much damage, and I do these just for demo purposes, using urea, because urea close to roots is pretty tough on roots at high concentration. So you can see that these roots and the seminal roots here got toasted, whereas this one put out one. But you'll notice that the shoots are coming up because that plant is living off the seed, and this is only like 10, 12 days old, is living off the seed. So even though the roots have been damaged, this one here, almost all of them, this one has popped out one, the shoot looks good. So if you're going to look for toxicity or any kinds of effect of nutrients on your roots, you're going to have to dig them up. Looking at the shoot isn't going to tell you what's going on at the root. Here's a close-up, and here's why. You can see that the tip is what gets damaged, because what did I say before? The tip has no protection. It's open. It wants to be monitoring things. So in this case, it's monitoring it, but it got it cooked. So you're going to see damage if it's going to occur usually at the root tip, or you might see it here if there's a granule there. So you're going to take a look, see at what's going on. Here's another example where I put urea here and it burnt off all the seminal roots. The shoot is fine. It's tight of burning, because what happened, these two remaining seminal roots are just putting out lateral roots like a darn. So the plant is compensating. And here's the last part of this presentation to make you think about your crop maybe a little differently, or maybe you're thinking about it. But we have two ways of looking at it. We have a source and a sink situation. So the source is where assimilates are produced, and then they're moved around, and then we have the sink. This is where the nutrients produced from photosynthesis, or the assimilates, sorry, from photosynthesis are moving to support growth. So where are those? Well, let me go back a bit. Remember this, the roots can't feed themselves. And if the roots need help, the shoot has to sacrifice and move more to the roots. If the roots are doing good and the shoot is doing good, you're going to get a maximum yield. If one or the other isn't doing well, maybe you damaged it with herbicide, or maybe you didn't get it up and going around, whatever. You did some damage to the shoot, then the roots aren't going to do well. And you can have your yield affected by a lack of source product or shortage of sink product. In other words, if you don't produce a good size seed because you were short during the growing season, eventually the sink, or that seed, is going to restrict your yield because you didn't produce enough cells in that seed to fill to make a really big seed. So you're going to limit there. You get what I mean? So you're always looking at what's the top doing, what's the bottom doing, what's the root doing? Are the leaves looking good? Is the root tip looking good? You know, am I in a drought situation? Are my roots flooded, which means they're not getting oxygen and they're struggling? If that's the case, the shoot has to move more roots nutrients down. Those are the kinds of relationships that are controlled by hormones, auxins and cytokinins and abscisic acid, all communicating with each other, that are reducing that yield. And the last two. So what are the sources? The seed is the source initially. So have a good seed, because that's what feeds that plant initially. Or a tuber. Have a good tuber, a well-nourished tuber. Remember what I said about the seeds that we tested with 2020 and the zinc deficiency and boron deficiency? We couldn't find that relationship, but when we put on a nutrient package on the seed, that helps to blend out some of those deficiencies. The leaf is the source. It produces the assimilates, which are used by everything. And that sinks, the seed, the roots, the flowers, and the seeds are really key. And if we don't have those built up to fill, we're in trouble. And here's the last slide for today. So how does the plant move that assimilate around. Well, here's what happens. We have the sugars produced here in the cell. It moves into something called a companion cell here next to the phloem. And this is a live cell. Phloem is live, xylem is dead cell tissue. Okay. Notice all these ports? Those are the plasmodesmata. So the plant moves it into companion cell. The solutes increase. They move it into the phloem. What do we know about where water moves when we've got a lot of solutes? High solutes. It moves from here in the cytoplasm, but from the xylem. There are ports in the xylem, so the phloem is pressurized by water, which forces it down. And why does it force it down? Because there's a negative pressure here, because it's being taken out here in the roots or somewhere else. So that water pressure moves it to the sink, and it unloads it at the sink. That's the root, the tuber, or it could be the apical meristem, because the leaves at the very top of the plant are in photosynthesizing. That removes the solutes, the water moves back, and the transpiration flow pulls it back up and recycles it. So now that you know, I don't know if you knew that, you know, for most of my life I wasn't a plant guy, but this is amazing to me, because now I understand why water not only affects, you know, transpiration and so on, it affects how it moves those photosynthates around in the plant, and why water is so critical, and why droughts are so devastating to plants. Anyhow, that's the end of this session. The next session will be about how plants deal with stress. And that's a really key one in terms of what nutrients are involved in stress management, and also how rhizobium and mycorrhizae form relationships with roots. Thanks a lot for attending. Hope you learned something, and see you in a couple of weeks.